Today we are going to be taking you through the process of meiosis in male Hieraciums, which are a plant that are more commonly known as hawkweeds. They have a diploid number of 8. We're going to start off in meiosis 1. Okay, so we're going to start off in the stage of interphase 1. So during this phase, all of the chromosomes and organelles are going to duplicate. So you can tell that the organelles are duplicated by the new centrioles. And in the chromosomes, we're now not going to have 8 chromosomes, but we're going to have 16. But these chromosomes will attach at the centromere to form a homologous pair. So we will now have 8 homologous pairs, or 16 individual chromosomes. Now that we have 8 homologous chromosomes, we're ready for prophase 1. So in prophase 1, you're going to start to see the chromosomes intertwining. The shape that they form is now called a tetrad. Once they're in tetrad form, it's time to begin crossing over. Crossing over is a completely random process, but it will determine everything about the resulting gametes. During crossing over, genes at the same loci from each of the chromosomes will break off and swap. This will create a combination of genes. So you could have your mom's dark brown hair and your dad's bright blue eyes. Crossing over is illustrated here, as you can see the genes breaking off and exchanging. This will eventually create 4 genetic variable chromosomes, but right now they're each combined in pairs. This will result in 4 different gametes at the very end of the process. And sharing of genes will result in genetic variation, which helps to strengthen us as a species. Now that crossing over is finished, our cell is ready to move on to metaphase 1. During metaphase 1, the nuclear membrane is completely broken down. Microtubules will extend from the centrioles, as seen here, to move our chromosomes to the metaphase plate, which is illustrated by the green broken line. The chromosomes will line up on either side of the plate, which is a process that is completely random and called independent assortment. This means that the way that the first pair of chromosomes falls will not affect the way that the second pair does, and so on. This will contribute to genetic variation. Now that the homologous pairs are lined up on either side of the metaphase plate, it is time to begin anaphase 1. During this stage, the recombined homologous chromosomes have genes from both mom and dad, or maternal and paternal genes. The microtubules, as illustrated here, will pull the chromosomes to opposite sides of the pole, so they are both moving closer to the centrosomes. So again, here we can see the microtubules that are pulling the homologous pairs back towards the centrosomes, and the We are now ready to begin telophase 1. During telophase 1, the chromosomes are already on opposite sides of the cell because of anaphase 1. The nuclear membrane reforms around the chromosomes, and the chromosomes condense back into their original chromatin form. As you can see from the shape of the cell membrane is beginning to furrow. After telophase comes cytokinesis. During cytokinesis, the cell membrane will pinch off, which will produce 2 genetically different cells. Each cell will have the recombined sister chromatids, which contain both maternal and paternal genes. Cytokinesis ends the process of meiosis 1, and we are ready to move on to meiosis 2. The first step of meiosis 2 is interphase 2. During interphase 2, the organelles will be replicated once more, but the chromosomes will not. Once interphase 2 is complete, it is time to move on to prophase 2. During prophase 2, the nuclear membrane will break down again, and the chromosomes will condense. Now that prophase 2 is over, we can now begin metaphase 2. Microtubules will line the chromosomes up, centromere centered on the metaphase plate. Again, we will see independent assortment as they are randomly oriented. This will again lead to even more genetic variation. Each sister chromatid will be facing the pole of the cell that it will eventually move towards. We are now ready to begin anaphase 2. During anaphase 2, the recombined sister chromatids will move towards the pole of the cell that they are migrating to. As they are moving, the microtubules will start to disassemble. It is now time for telophase 2 and cytokinesis. During telophase 2, you will see the nuclear membrane reforming around the chromosomes, and during cytokinesis, the cell membrane will pinch off, which will leave us with 4 new sperm cells. Each sperm cell will contain a different variety of genes from both its mother and father, which will contribute to genetic variation in the eventual offspring.